All right, we'll go ahead and get started. Thank you all for being here today for the second of Pennsylvania 4R Alliance's Fall Webinar series. My name is Brian Campbell from the Nature Conservancy, which is a member organization of the Pennsylvania 4R Alliance. And I will go ahead and post in the chat some important information. So there in the chat, you will find a link to the Mid-Atlantic 4R Association, which is our parent organization, and then some additional information I'll talk about in a moment. So the Pennsylvania 4R Alliance exists to promote the 4 Rs of nutrient stewardship, and those are applying the right crop nutrients or the right source at the right rate, right time, and right place. To illustrate what that looks like in practice, our organization developed a 4R scorecard for the Mid-Atlantic, and you can find that link posted near the top of the chat box. I think that's link number 2, or the item number 2 in the chat. The Pennsylvania 4R Alliance exists to promote the 4 Rs on Pennsylvania farms through research, education, advocacy, and collaboration. And you can see here that there's a broad coalition that's actively furthering that aim. Dr. Charlie White is a key partner in our work. He serves on the board of the Alliance and is our speaker today. Dr. White is assistant professor and extension specialist of soil fertility and nutrient management at Penn State University. His extension, research, and teaching work focuses on efficient and effective use of ag nutrients. And today he'll be updating us on a decision support tool that he and his colleagues have developed. We'll have time for questions at the end of this session, but feel free to enter questions in the chat as well. For those of you interested in continuing education credits, I will post a link to a sign-in form at the end of the session. And finally, I would like to thank and acknowledge Northeast SARE for their support of this fall webinar series. With that, I will pass things over to Charlie.
Great. Thank you much, Brian. Thank you very much, Brian. It's great to be here today and to be back with the PA4R Alliance for another webinar series. I appreciate all of you attending today and supporting the Alliance in your day-to-day activities by spreading the good word about the 4 Rs and by supporting the organization by attending this webinar and supporting your own professional development with this webinar. So let me bring up my slides here and we will get started with the presentation. So I think some of you have probably heard me talk before over the last couple of years about some work that we've been doing to try and update and improve nitrogen fertilizer recommendation method. And so this is kind of an update on the ongoing work. It has been a very successful project so far, and since the last time I've shared this work with folks, there might be some new developments in it that I wanted to share. And for those of you that haven't heard about this work, maybe this is your first time hearing about it, then I'm hoping that there'll be enough background to bring you up to speed in what we're working on with this. So you all know that nitrogen is a very complex nutrient. It's a complex element to work with. It's got lots of different forms in the soil and in the atmosphere and in the water. It's always changing forms. It's a very dynamic element. It's changing forms because of microbial and biological processes. And we are not in charge of these microbes. They march to the beat of their own drum, and they're really dependent on the weather. And so we're just sort of managing around what they do to the best of our abilities. There's lots of loss pathways for nitrogen. And when it does get lost into the water or get lost into the air, it tends to be an environmental pollutant. And so we're concerned about managing it from that perspective. It's also one of the most expensive, probably this year it was the most expensive crop production input for nitrogen demanding crops. And so we also, from an economic perspective, want to steward it as wisely as possible and use only enough as what we really need to get a profitable crop. So, in tandem with trying to improve nitrogen management, a lot of farmers are also trying to improve soil health, and they're implementing soil health management systems, we could call them. So reducing tillage and integrating cover crops. And these are really great practices for a number of reasons. But one of the challenges that comes along with that is that they can dramatically complicate nitrogen management, right? Because we start growing cover crops, and lots of farmers are choosing different types of cover crops. They might be growing legumes, like here's a picture of a red clover cover crop and a crimson clover cover crop. Here's a cereal rye cover crop that's been grown to maturity and killed and then planted into directly planting green, right? And so depending on the type of cover crop you grow, or maybe you're growing a mixture, how you manage it, it can really complicate nitrogen management, because sometimes these cover crop residues might immobilize nitrogen, tie it up, and you might need to apply more fertilizer. Other times they might mineralize nitrogen, and you could apply less fertilizer. Over the long term, a big heavy rye cover crop like this that produces a lot of biomass over the long term, that's going to increase soil organic matter levels. But in the long term, that's a good thing. Probably after 8-10 years of doing this, you're going to have higher soil organic matter, and you can pull back on your nitrogen. But the first year you try this, you might need to increase nitrogen, right? And so how do we develop a system that accounts for all of this diversity of management on different farms, a system that everyone can kind of use and take site-specific measurements of their soils and their cover crops and integrate them into a recommendation. So that's really what we're trying to get at. We've been doing a lot of on-farm experimentation. This slide shows you from last year, the 2021 growing season, shows you 6 different experimental locations on different farms where we did nitrogen fertilizer response trials, and you see 6 very different response curves, right? They differ in what their yield plateau was and what the nitrogen required to get to that yield plateau is. And the variation in these response curves comes from their cover crop management this year, but also their historical practices, their soil organic matter levels, their manuring history, all of those things. So there really isn't a one-size-fit-all type of recommendation, and these on-farm experiments really confirm that. So we've been developing this tool over a number of years, and it really has been a community effort. We've done lots of on-farm research, and these are some of the collaborators that have donated their time and management expertise and land to help with this. A number of students and postdocs and technicians and research and extension staff and funding have contributed to this. So I want to recognize that early on, that it's really been a community effort, and it's thanks to all of these participants that we've gotten as far as we have. So one of the things that we started to understand as we were delving into this is that the driving factor for nitrogen requirements of corn were a little bit different than maybe we have come to understand or been taught in the past. And, a lot of times in the past, we have thought that the nitrogen fertilizer requirement is tied to the yield goal, right? So 1 pound of nitrogen per bushel of corn. But here are 2 response curves from 2 different sites in the same year, but they had very different weather patterns. So in Center County, we had a severe drought, and our yield only reached 113 bushel per acre because of that drought. But it still took us 173 pounds of nitrogen to get there, right? So that definitely violates our 1 pound of N per bushel of corn requirement, right? Here's another response curve that we got 214 bushel per acre and only needed 172 pounds. And so that's maybe a little bit more in line with the 1-to-1 requirement. This site did have soybeans as a previous crop, so you could take a little bit of a credit for those soybeans, and maybe that would bring you down to a recommendation of 172 pounds of nitrogen. But what is it about these response curves that is the same that would maybe indicate they both needed 173, 172 pounds of nitrogen? The thing about them that's the same is this characteristic that we call the delta yield. And it's the difference between the yield in an unfertilized condition with 0 nitrogen, which is what the soil can supply alone, versus what your yield plateau is given your environment, your soil, your growing season, your management skill. That differential is called the delta yield. And these two response curves have the same delta yield of 40 bushels per acre. And it's not a coincidence that they both needed the same amount of nitrogen fertilizer. Here's another example of two different response curves. Here we have Berks County after a mature wheat cover crop, so very high biomass, high immobilizing potential of this site. And we see that to get 250 bushel corn, we needed 275 pounds of nitrogen. And the reason for that large nitrogen requirement is because of this large delta yield. We basically have lots of immobilization pulling this response curve down to a very low unfertilized yield. It's a great environment, great soil, great growing season, and Berks County that year produced 250 bushel corn. But to cover that large of a delta yield there, to get from our unfertilized condition to our yield goal, required a lot of nitrogen fertilizer, 275 pounds of N. Here's another site, Lancaster County, where similar yield plateau here, 241 bushels, similar to kind of Berks County. So again, a good soil type, good growing season. But our unfertilized yield is fairly high. And this is a site that has a heavy manure history, and they grew a rye cover crop that they killed when it was fairly young and probably mineralized lots of nitrogen. It's got a small delta yield here, only 30 bushel per acre. And so it only took 134 pounds of nitrogen to cover that relatively small yield gap. So looking at lots of these response curves, we started to understand that this delta yield was the property that was really driving the nitrogen fertilizer requirement. And this is kind of a busy chart, but what it shows is on the x-axis here is the delta yield. And this is research data, so we've got it in scientific units of megagram per hectare, but there's about 19 bushels per acre per megagram, in case you wanted to do the conversion. And then on the y-axis here, we've got the nitrogen rate to get to the yield plateau. So how much nitrogen was required basically to get to your yield goal. And there was a strong and predictable relationship between the delta yield and the nitrogen requirement for the yield plateau based on the region you were in. So you can see we've got data, and this is a big national project that we led to compile a bunch of response curves from different states. So we've got Delaware, Maryland, Pennsylvania, Illinois, Iowa, Nebraska, Kentucky, Virginia, Wisconsin here on this chart. That's why it's so busy. But each of these regions has a relationship, an underlying relationship that we statistically determined that can be used in the future, right? So it's interesting, Maryland and Pennsylvania being right next to each other, they fell on the same line, this blue line. Illinois and Iowa being next to each other, regionally close, similar soil types, they also fell together on this green line, which, and so each region has a slightly different ecosystem, soil ecosystem, agricultural ecosystem that sort of changes this relationship slightly. But we can use this in a tool to develop nitrogen fertilizer recommendations if we can find a way to predict the delta yield. So how do we go about predicting that delta yield? Well, mathematically, it's a simple formula. You take your maximum attainable yield and you subtract your yield with 0 nitrogen fertilizer applied. okay, that's simple math, but how do we predict the maximum attainable yield? How do we predict the yield with 0 and fertilizer applied? That's a little bit more challenging. Maximum attainable yield, that's going to be dependent on your climate, your soil type, your corn genetics, your management skill. It's going to vary from year to year based on the weather, that's for sure. But it's probably going to vary within reasonable bounds, and you've probably got some experience, you or the farmers you work with, probably have some previous experience keeping track of your yields that you could determine typical range of yield goals, determine an average yield goal for your scenario, for your context. That would be your maximum attainable yield. So I think we can get this pretty easily based on prior experience growing corn in different fields on a farm. Now, the yield with 0 nitrogen fertilizer applied, that's a little bit more complicated. This is dependent on the soil nitrogen supply. It's dependent on the mineralization rates, how much soil organic matter you've got built up. What's your cover crop nitrogen content, your cover crop C to N ratio. It's dependent on some of the microbial physiology that's breaking down these materials and mineralizing nitrogen. It's dependent on some things that we understand from knowing about this process and studying the microbial physiology. You know, people have been studying this process for decades. There's a lot that we actually do know about it. We know that it depends on temperature and moisture, so your climate. We know that it depends some on soil texture. It depends on the organic matter level and the cover crop nitrogen and C to N ratio. And so the question is, with our current level of knowledge about how this process works, is this something that we can predict? And I put question marks here because a lot of people think it is hard to predict, but with what we know about it, we can predict this. And that's kind of the basis of what our tool has developed around, is how do we predict this unfertilized yield based on an understanding of the nitrogen mineralization process. So let's talk about that nitrogen mineralization process a little bit and understand the controls that are at play so that you understand what we've brought into our tool in terms of being able to make this prediction. So in this diagram here, we've got a decomposing organic material. That could be your native soil organic matter. That could be a cover crop residue. And let's just leave it at those two things for this point. You know, could also be manure additions or previous corn stove or soybean residues, right? Those are also important, but for this point, what our tool is measuring here, or what we're predicting, is from soil organic matter and cover crop residues. Those decomposing organic materials are going to have a component of carbon and a component of nitrogen, and we can speak about them. The characteristic of that relative ratio is the C to N ratio. So if you've got a lot of nitrogen in your material relative to the carbon, that's going to be a low C to N ratio. Okay, then we've got the microbes. They're the main actors here in this story, and their biomass is also made up of a certain ratio of carbon and nitrogen. On average, microbes tend to have a C to N ratio of about 10 to 1. So for every 10 parts carbon, they need 1 part nitrogen to build their biomass. Okay, so what are the microbes after, right? What they actually want is to break down this organic matter. They want to break down the carbon, and they get energy from the carbon to fuel their metabolic activities. And when they get energy from that carbon, they respire that carbon back into the atmosphere as CO2, right? Basically, as they break down that sugar molecule and the organic matter that they're eating, they get energy from it. They breathe that energy back into the atmosphere as carbon dioxide. And the other thing that they're doing is they are using some of that carbon to build their biomass, right? They want to grow a bigger cell, and they want to divide, or they want to lengthen their fungal hyphae through the soil. And so the proportion of carbon that the microbe uses to build its biomass versus what it uses to generate energy and respires that into the atmosphere as carbon dioxide is called the microbial carbon use efficiency. And that can vary. So the efficiency is how much they build into their biomass. That can vary from anywhere from 25 to 50%. Is typically what people think, but in our tool, sometimes we see that vary, up to 75% sometimes building carbon into their biomass. Now, why is it important to understand how much carbon they build into their biomass? Well, it's important to understand that because with that carbon, they also need to take up nitrogen. And so if we've got nitrogen decomposing from our cover crop residue, we need to understand how much of that nitrogen is going into the microbial biomass. And that nitrogen going into the microbial biomass is tied to the carbon that's going into the microbial biomass. Okay, now let's say we've got a decomposing material with a relatively low C/N ratio, means lots of nitrogen in that material. Microbes take up what they need based on the carbon that they took up. And then there's possibly a little bit of nitrogen left over from the decomposing material, and that gets turned into ammonium and released into the soil. And that is what we call the mineralized nitrogen, ammonium being a mineral form of nitrogen or an inorganic form of nitrogen. And that's what can feed our crops, right? So if we understand some about this process, if we understand what's the microbial carbon use efficiency, if we understand the amount of nitrogen in the cover crop residue and the C/N ratio, we can predict this amount of mineralizable nitrogen. And there's a, it looks like a kind of hairy equation here that, predicts that, but that's what we can use, right, in our tool. The amount of nitrogen in the cover crop, the amount of microbial carbon use efficiency, and the C/N ratio of the cover crop and the C/N ratio of the microbes. This equation helps us, and K here is a rate of decomposition. This helps us predict the amount of nitrogen that's mineralized from some measurements of the cover crop residues. Okay, so this same process can occur, but sort of in reverse, what we call immobilization, if the cover crop has not enough nitrogen in its biomass. So here I've shrunk the amount of nitrogen in the cover crop, and it's got a high C/N ratio now. As microbes decompose this, they build their biomass with carbon. They need a certain amount of nitrogen, right? This green arrow here is how much nitrogen they need to build their biomass. Well, the cover crop residue doesn't have enough of it. And so where are they going to get that nitrogen from? They get that from inorganic nitrogen, either ammonium or nitrate that's already in the soil. The microbes are going to take that up. And when that process happens, what we're actually doing is we're drawing down the available nitrogen in the soil, and we're taking it away from our crop to feed the microbes. And when we do that, we need to apply more nitrogen fertilizer to make up for that process. Now, is this immobilization process necessarily a bad thing? Well, in 1 year, it might be, because it requires us to apply more nitrogen fertilizer. But that nitrogen isn't being lost from the system that's being immobilized. It's being built into microbial biomass, which will then turn into soil organic matter. And then that soil organic matter in future years might be the new pool of carbon and nitrogen that could mineralize and become available. So over time, if we're immobilizing nitrogen and building it up into soil organic matter, that's sort of like taking some of your money and putting it in a long-term savings account that pays interest on that, and it will come back in the future as a credit to you. Okay, so some of the key characteristics of the cover crop residue that controls nitrogen mineralization is the C/N ratio, but also the total N content, right? So we've got the C/N ratio, the cover crop up here, we've got the total N content of the cover crop over here. And at some point, the C/N ratio gets high enough that you reach a tipping point where immobilization starts to occur. That tipping point is often around 20 to 1, but it can be as low as 15 or 16 to 1. It could be as high as 25 to 1. And one of the things that controls this is the clay content of the soil. So when we get to our tool, you'll see that the soil texture, the clay content, is one of the inputs to the tool. And that controls exactly where this sort of threshold is that cover crops turn from being mineralizing to immobilizing. But we have different cover crop types. Clovers and peas that are legumes tend to have low C/N ratios, and they're generally fairly reliable in terms of being mineralizers. The one exception to that in the clover world is crimson clover. And a lot of people like to grow big crimson clover cover crops where the flowers come out and it supports the pollinators, and you've got lots of biomass, and you've got good soil coverage, for erosion control. But crimson clover, I have seen that its C/N ratio, as it gets more and more mature, creeps up into territory where it's no longer actually releasing that nitrogen back into the soil. Doesn't necessarily immobilize nitrogen, but it's sort of a net neutral effect in terms of its release of nitrogen. We've got radishes that winter kill in the fall, tend to be fairly low on the seed-N ratio. Canola, another brassica. Depends how long you let it grow and how mature it gets. It can certainly grow mature enough to become stemmy enough and have a high seed-N ratio and tie up nitrogen. And similar things happen for grasses like rye, triticale, wheat, annual ryegrass. If they're killed when they're very young, they can have a low seed-N ratio and mineralize that nitrogen back. I have also seen that some grass cover crops, for example, triticale, this year we had an experience where swine manure was applied to the triticale and it was injected, and the triticale got very tall, but because of all of that nitrogen in the swine manure, it still maintained a low C/N ratio. But generally, if you're not, unless you're fertilizing your cover crop, if you're not fertilizing your cover crop with manure, then typically as the cover crop gets more and more mature, its C/N ratio starts to creep up and potentially become immobilizing. And then mixtures can really run a wide range. It depends what's in the mixture, the species composition, how mature that mixture gets, and whether manure has been applied to that mixture or not, or there's lots of leftover nitrogen from the previous year. That can really, influence the seed-N ratio. Okay, so nitrogen content being an important characteristic of the cover crops, how do we go out and measure nitrogen content in the field, right? Do you go out like a scientist like me and get down with a quad rad and clip that biomass and put it in a paper bag and dry it and then weigh it and send it to the lab? No, you probably don't want to do that, right? So we recognize that, and there's a tool that's available that agronomists were starting to purchase, a handheld Greenseeker here that was designed to be walked over wheat or designed to be walked over corn and measure the greenness of that wheat or the greenness of that corn. Well, what it's really measuring when it's measuring that greenness is it's measuring the nitrogen concentration and it's measuring the density of biomass in that wheat or corn crop, and it's giving you a number, the NDVI value from 0 to 1. So what we thought was, well, we could probably walk this over cover crops, and it's probably going to tell us about the nitrogen content of the cover crop biomass. So we walked it over hundreds and hundreds of cover crop plots that we were also clipping and drying the old school way. And we found fairly strong relationships between the NDVI value and the biomass nitrogen. And so this is now part of our tool. You could walk out to a field and walk this Greenseeker sensor over the crop. Here's a picture of one of the students in my group, Zach Sanders, walking this over a rye cover crop in the spring. And you can see the value that's showing up here. And you can take that number and convert it into pounds nitrogen per acre of your cover crop biomass. For those of you that don't want to go out and walk every field in the spring, we're also looking at ways that you could use satellite imagery to do this, because satellites, they're measuring NDVI just like this handheld sensor is, except they're doing it from outer space. They're doing it over the whole planet. They're doing it every 24 hours. So if you've got a cloud-free day, sometime around when you're going to be killing your cover crop, and you've got a satellite, subscription to satellite imagery, you could probably calculate nitrogen content of your cover crops on all your fields without even leaving the office, is sort of our vision for the satellite imagery. But we need to work on that. That's sort of a next phase of the project. Okay, so behind the scenes of this tool, there are a bunch of hairy equations that we use to predict unfertilized yield. The first thing we do is we calculate the humification efficiency or carbon use efficiency, so the amount of carbon that the microbes are going to retain. And that influences mineralization from soil organic matter and cover crop residues, as we talked about. Next thing we do is we look at the corn yield response to nitrogen mineralization from soil organic matter. So we have the soil percent carbon that we measure from a soil sample. We calculate the corn yield response to nitrogen mineralization from cover crop residues. So here's our little equation that I showed you before. We've got the N content of cover crops. We've got the carbon use efficiency, which we calculated up here from soil texture. We've got the C/N ratio of the cover crops. And we've got a parameter here that we have calibrated to our field experiments so that we can predict based on the cover crop, inputs, what is the unfertilized yield and how that's affected by the cover crop nitrogen and seed-N ratio. So we've got a component of the yield credit from soil organic matter, a component of the yield credit from the cover crop residue. We add those together and we observed that there was kind of a quadratic curvature to that yield response. Just like when you add increasing amounts of fertilizer, there tends to be a quadratic curvature to the yield response. Same thing happened with nitrogen from cover crop and soil organic matter, right? That kind of makes sense because those things, when they mineralize, they're mineralizing it as ammonium. It's not that different than adding increasing doses of urea to the soil or ammonium sulfate. So it kind of makes sense that it follows a similar biological response with that quadratic curvature. And so we found from an initial calibration data set that we got a pretty good relationship between the predicted grain yield and the observed grain yield. And again, these are in scientific units from the research paper that we wrote looking at this. Okay, so how do we use this tool, this prediction of unfertilized yield, in our decision support tool? The key thing is that we are predicting the unfertilized yield from mineralization of nitrogen. We have a yield goal that we know from previous experience growing corn in our climate, soils, and management systems. And from that difference between the unfertilized yield and our yield goal, we get the delta yield. And then we can look up on that relationship I showed you for Pennsylvania and Maryland. They fell on the same line. There's a amount of nitrogen needed to close that yield gap, or close that delta yield. So the neat thing about this system is we can make predictions of unfertilized yield for, say, a rye cover crop or no cover crop or a legume cover crop. And we would expect those delta yields to change based on the mineralization of nitrogen from the inputs. And that change in the delta yield changes our fertilizer requirement. And we can look at that with low soil organic matter, medium soil organic matter, high soil organic matter. That's going to change the delta yield. Or different soil textures, right? All of these things actually end up interacting with each other in ways that you couldn't really guess until you have the decision support tool, but they all interact with each other to affect the nitrogen fertilizer recommendation. So we've been working on this tool for several years. It's been through a number of iterations. On our website, we have the very original tool that was developed from a paper that we published in 2020, which meant the data was collected in 2018, 2019. So it's kind of old, but we put it out on the website for folks to try and experiment with as a learning tool. But since then, we've evolved. We've continued to do more work. We've integrated that work into the equations behind the scenes. And we've got a version 2 of the tool. And the version 2 of the tool has some changes to it. The first thing is that what it actually draws is it draws a full quadratic response curve, basically connecting your unfertilized yield to your yield goal and determining the maximum nitrogen that's needed to reach that yield goal. Drawing a quadratic relationship there allows us to do the economics of fertilization. So you can see where exactly on that quadratic response is the economic optimum, where the next unit of nitrogen that you apply is going to cost more than the yield response that you get, right? That's the point. The economic optimum point is the point right before that last unit of nitrogen doesn't pay for itself. This is a little video here. It shows you how you can interact with the tool. So you can set your yield goal, and as the yield goal increases, the nitrogen fertilizer required to reach that yield goal also increases because your delta yield is increasing. As you change the sand content, it changes the carbon use efficiency, which changes the mineralization rates, and that changes your delta yield. Increasing the organic matter means more mineralization, changes the delta yield. Adding cover crop nitrogen increases your unfertilized yield, reduces your fertilizer requirement, unless you've got a really high seed-N ratio. Here we're increasing the seed-N ratio, and we see that yield, unfertilized yield, really dropped down. And then finally, we can adjust the cost of nitrogen fertilizer or the value of grain and see that our optimum fertilizer rate is changing based on the economic scenario. And, that type of power in a tool would be very useful in today's marketplace where we've got expensive nitrogen fertilizer, right? How much should you be dialing back on your rate because of nitrogen costing $1 a pound, right? So the tool allows you to look at that. Okay, so that was version 2 of the tool, where we went to sort of this type of interface with the response curve. We're on to version 3 now, based on the most recent years of on-farm experimentation, where we've sampled more different types of soil textures, more different types of cover crops, different yield goal or yield attainable yields in different regions. And we've got version 3 now. And there's a couple nuances here. First of all, our soil texture control changed from sand to clay content. Next, instead of just measuring the crude organic matter, as you might have historically done on a soil sample, we're finding that it's more accurate to get a true analysis of total carbon and total nitrogen in your soil, rather than just the crude measurement of loss on ignition, where you heat the soil sample up in a furnace and measure how much weight is lost. There are instruments that can actually measure the true amount of carbon and the true amount of nitrogen in the soil specifically. And that percent carbon and the soil C to N ratio help to make for better predictions of the unfertilized yield. Winter killed cover crop nitrogen, spring cover crop nitrogen, spring C to N ratio, all still the same and important variables. And then price of corn and price of nitrogen are in there. So the main changes are the soil texture and measuring soil carbon and soil C to N ratio specifically. What I showed you there was the graphical interface, but we've also got this programmed into a spreadsheet, so you could do bulk recommendations, right? I know the last thing you all want to do if you're making recommendations for farmers is to fiddle around for each field with a little slider bars like you just saw in that video, right? Instead, it'd be nice if you had a spreadsheet where you could just drop your soil test values in, drop your yield goals in, and then have a column that has nitrogen recommendations for every field. So we've got a version of the tool in a spreadsheet as well. Okay, what I want to look at here as we wind down a little bit are some examples, on-farm examples of how the tool is working, just to give you some illustrations. So this is a site from 2020, and there was no cover crop grown there. We plugged in the corn yield goal for the site, the clay content, soil carbon, soil C to N ratio, and we got a response curve that looks like this. And based on corn and nitrogen prices in 2020, $4 a bushel corn and 30-cent nitrogen, it was calling for 161 pounds of nitrogen as the optimum rate. Our experimental response curve was showing 172 pounds of nitrogen required. And this 172, there's sort of some error bars around there, and removing any one of these points that might have been an outlier might shift that a little bit. So 161 pounds from the tool compared to 172, to me they are basically equivalent, and it's going to be hard to get nitrogen recommendations that are accurate more closely than these are to each other, right? I think they're 11 pounds nitrogen different from each other. Here's another scenario from 2020. This is where we grew a mature wheat cover crop, or the farmer grew a very mature wheat cover crop. And you can see how much this response curve drops off here without any nitrogen fertilizer applied. Because of the immobilization from that wheat crop, he would have only been producing about 50 bushel corn. You can see the high C to N ratio here, 33, and relatively high nitrogen content, which basically just means there's a lot of biomass. Lots of biomass with a high C to N ratio means lots of immobilization. One of the outputs from the tool shows you actually this cover crop yield credit. It shows you the effect of the cover crop compared to having not grown a cover crop at all. So basically, compared to a fallow field, this cover crop immobilized 90 and dropped the yield 92 bushels per acre, right down here on the unfertilized yield, because of that cover crop residue. We can also see the critical C to N ratio for immobilization, like the point where it tipped the seed-end ratio would have tipped from being mineralizing to immobilizing was as low as 14:1 in this scenario. So here we've got a seed-end ratio of 33, and that's really affecting the drop-off with this yield response. So this is what the tool predicted based on its calibration, and the response curve at the site was fairly similar in terms of its sharp level of drop-off and its requirement of 275 pounds of N to reach that yield goal. Again, 275 within margins of error is very close to what the tool is recommending of 257 pounds of N per acre. So when we've looked at using this tool across a number of different farm scenarios, and we look at what the tool predicted as the economic optimum nitrogen rate and what we saw from the experimental results as the observed economic optimum nitrogen rate, we see that there's a nice relationship, and it's very close to the 1-to-1 line. So 150 pounds predicted here, 150 required as being observed there, 200 required, 200 observed. So we're very close to that 1-to-1 line. There is some scatter around the line, so it's not perfect, but it's trending in the right direction. And it's, I think, probably, within, a complex nutrient like nitrogen, it's going to be hard to get closer, I think, than this, although, we're always going to be striving to get closer and better. But right now it's faring pretty well. So one of the things we did observe as we looked at these relationships is the tool seemed to work the best when the yield goal was input as a multi-year average for the site, rather than plugging in what your yield plateau was in any particular given year. So, and that's actually really helpful to know that the tool works better, because you can't predict what your yield goal is going to be in any given year when you have to make a nitrogen fertilizer recommendation. So much of the growing season is still ahead of you. But knowing that the tool actually works better based on your average yields is great, because that is something we can keep track of, and you can know ahead of time what your average yield is for a field or a site. One of the reasons that we think the tool works better for using the average yield is, for example, a year with good moisture, you get a higher than typical yield because of that moisture. You might think, oh, well, I need more nitrogen because of that higher than typical yield, right? A greater nitrogen demand. But in that good weather year, there's also good soil moisture, which promotes more nitrogen mineralization. And so your unfertilized yield probably went up as well, and the delta yield probably stayed the same. And the converse would be true for a poor year, right? Very dry year reduces your yield goal, probably reduces your nitrogen requirement, but it also reduces mineralization rates, and probably your unfertilized yield is sinking as well. So basically what I'm trying to say is that response curve in different years is just sort of moving up and down with the weather. It's not really changing the slope or anything like that. And so that's why the tool that is calibrated for average yields works best when you put in, or the tool being calibrated for the average across many growing seasons works best when you put the yield goal in as the average of many growing seasons for your yield goal, if that makes sense. The tool is really useful in terms of potentially doing some scenario building. So here's some scenario testing where this is the same site that grew that mature wheat cover crop, right? And required 257 pounds of nitrogen. That was 2020 corn and fertilizer prices. What about this past year, where corn was running about $6 a bushel and nitrogen was $1 a pound? How did that affect where the economic rate is on this response curve? Well, it pulled it down from 257 pounds per acre to 241 pounds per acre. That makes sense because nitrogen is more expensive, but the value of the crop is also more, and that tends to push the fertilizer rates up. So we did see on net, the net effect was a slight drop in the fertilizer, but it was fairly small, right? 16 pounds per acre difference, right? So we're not talking about cut it by 100 pounds. We're talking about cut it by 15 to 20 pounds of nitrogen, given the differences in economics. You could also use this tool to say, hey, you know what? 2020, I grew a mature wheat cover crop. Fertilizer was cheap. Why not do that to build soil health, increase organic matter? right? Here we are, or here we were in 2022 with $1 a pound of nitrogen. You're thinking, should I let this cover crop grow or should I kill it now when it's young? And you could use this tool to do some scenario building. So here we looked at reducing that seed-end ratio from 33 where it was previously to 15. And that's also killing it when it's young. It's not going to have as much nitrogen in it either. So I dialed the nitrogen content down to 40, which is kind of typical for what I've seen in a grass cover crop killed when it's still vegetative. These are pretty typical values. Previously, I think I pointed out that cover crop yield credit was 92 bushels per acre when it was killed mature. Now it's only -5 bushels per acre when it's killed vegetative. And so that brings up this unfertilized yield here, and it pulled down our nitrogen fertilizer requirement to only 193 pounds per acre, right? So you could use this tool in any growing season to say, hey, you know what? Nitrogen is really expensive this year. Maybe this isn't the year to grow a big, massive cover crop. I'm going to kill it when it's young, and I'm going to save myself $48 an acre by having 48 pounds less nitrogen required to reach my yield goal. Now, we want to talk long-term, and I discussed this with a lot of farmers over the summer, and they said, you know what, I'm committed to soil health. I'm committed to growing big cover crops. I'm committed to increasing my soil organic matter. Yeah, I know this is a tough economic market, but I'm going to stick to my guns and I'm going to grow my big cover crop. And over the years of doing that, I'm going to expect my soil organic matter to go up. So I wanted to run another scenario where this farmer stuck to his guns and increased his soil organic matter from 1.4% to 1.9%. And what we saw is that had such a big effect on nitrogen mineralization that even with a mature cover crop here, 33 C to N ratio, and a big cover crop yield penalty here, because of growing that big cover crop over time and potentially raising soil carbon levels to a fairly high level, we saw that overall that effective building soil carbon had a bigger impact in terms of reducing fertilizer by 55 pounds nitrogen per acre, and still growing that big cover crop, right? So that's really the impact of growing soil organic matter, putting it in, or growing cover crops, building soil organic matter, putting that in that long-term account that's going to pay you interest on it. That's where we saw a big effect. So again, the tool can be used for scenario development like that. So just in concluding, before we stop to take a few minutes, have a few minutes for questions, this is a tool that we developed, and one of our primary goals was to be able to account for the underlying biological processes that affect nitrogen supply to cover crops, primarily that microbial mineralization process. By basing it on the biology, we create a robust tool that is now able to accommodate a wide variety of practices, right? Different cover crop types, different C to N ratios, different management styles of when you terminate it. All of that diversity is represented across the agricultural landscape and can be accommodated by this tool. I want to recognize again that it was a community effort to get this done and get this tool built, right? We never would have been able to calibrate it if we just had a really narrow range of conditions. If I had just worked at the research station and not explored the diversity of soil types and management systems in the agricultural community, we wouldn't have been able to calibrate this tool. So I'm really appreciative and thankful that for all of that community effort, and members of the PA4R Alliance have been part of that by funneling some of their clients into our on-farm testing program. We've been able to, that's one of the ways we've been able to do that. So I welcome other members of the PA4R Alliance and others who want to funnel farmers into our on-farm testing program to help refine this. I want to encourage you to keep doing that. And lastly, I want to say that this is a recommendation framework that's a platform. It's not an end product. It's a platform that we can continue to adapt and improve. Some research that we're working on are figuring out ways to get better predictions of microbial carbon use efficiency. There have been a couple sites that are just real oddball sites. They're not being predicted well by our tool, and we think it might have to do with something unique about the microbes there. It mentioned satellite imagery to calculate cover crop nitrogen content. I think that might be a new frontier in sort of implementing this tool. And a lot of folks have said, you know what, it'd be nice if I had a cover crop C to N ratio lookup table, right? So that I could plug those values in based on growth stage of cover crops or species types. So that's something we're working on as well. So just again, thank you to all the people that have contributed to this, all the on-farm collaborators, students, technicians, other research staff, and funding agencies. It's been a great success, and I'm glad to have been able to help lead this. So Brian, I'm going to let you moderate any questions here. So I'll turn it over to you and see what we have.
Sure. Thank you, Charlie. I don't see any questions in the chat. If anyone would like to, oh, I'm sorry, it looks like Rita just posted. You might have mentioned this, but were legume cover crops part of the field test?
Yes, yeah, we have had legumes. We've had some red clover monocultures. We've had this past year we had a hairy vetch field. We have had trying to think some of our other legumes. We've had crimson clover. So yeah, we have had legumes. Not as many pure legumes as we have had legume grass mixtures. So we've had lots of legume grass mixtures in the experiment, but fewer pure legume monocultures. Yep.
And Nicole asks, can you post a link to the tool, or where can we find it?
Yeah, so our current tool is version 1 of the tool. If I back up my slides here, which I think are still being shared, got version 1 of the tool. There's the link here. I'm in the process of getting version 3. Version 3 of the tool loaded onto our website. So it'll be at that same link though. Yep.
Great. And I don't know if you prefer me to read them, but Eric asked, why does the delta yield require a significantly higher amount of nitrogen per bushel of yield?
Yeah, so that's a great question. So that per nitrogen per bushel of yield might be 2 to 3 pounds of nitrogen per bushel. And that has to do with a couple of things. One is we're now looking more at like the physiological requirement to put additional units of nitrogen into the grain and additional units of nitrogen into the stems and the leaves and things like that. And so if you basically look at that nitrogen required to grow that grain plus the stem and the leaves, it's higher than just for the grain, right? So like a bushel of grain might require 1.2 pounds of nitrogen to grow that grain, but you've also got to grow the leaves and grow the stems. And this also accounts for some level of inefficiency, some level of losses. But basically it's also, we're saying we're counting just for that gap, not for the whole amount, right? So the one pound of nitrogen, or some people use 1.1 or 1.2, that's for the whole amount. We're saying just for that gap. So you end up with a bigger number multiplied by a smaller gap. In the end, it actually comes out somewhat close to our traditional recommendation of doing it on the whole, but doing the gap allows us to look at the variations in that unfertilized yield.
It's super helpful. We had several people interested in the geography limitations of the tool. Can it be used on our bigger farms in Eastern Virginia, on the Delmarva Peninsula? Where do you see the geographic limitations of this tool?
Yeah, so the geographic limitations come into play when we are calibrating the coefficients in the model. Those need to be calibrated to specific regions, right? So I showed you that nitrogen required to cover the delta yield, all those different slopes of those lines for each region, it's slightly different, right? And so that's an example of us calibrating that component of the model to all these different regions. The other part of the model is the prediction of the unfertilized yield. That also needs to be calibrated to different regions, but it's relatively simple types of experiments that can be done to do that. It's basically just growing corn without any fertilizer and measuring the yield and taking a soil sample so you can get the texture and the carbon and the nitrogen and a sample of the cover crop biomass. And I'm actually working with collaborators in Virginia now, a graduate student there, with Wade Thomason has collected and Rory McGuire has collected lots of historical data where they actually have the data they needed to calibrate the model. And so we actually, just this past weekend, I calibrated the model for Virginia and it worked. It's got slightly different coefficients than Pennsylvania, but the underlying theory is the same. The biology is the same, just needs slightly different coefficients.
So as far as this existing tool, you feel that it's solid just for Pennsylvania?
Yeah, the numbers that come out of the current tool are only solid for Pennsylvania. We would need to do a recalibration for Maryland or Virginia to get those numbers, but it's relatively simple to do that. And again, I think the tool is a platform, it's a foundation that we can build on and do these, state-specific calibrations.
Yeah, it's great to have that to build off of. Eric also asked, what growth stages should cover crop NDVI be performed on?
Yeah, so ideally you would get the NDVI as close to cover crop termination as possible, because the cover crop will continue to grow and continue to take up nitrogen, and that NDVI value will probably increase over time. So if you are, if you're going to take a ground measurement with a Greenseeker sensor, you could walk the field the day you terminate, and that would be the most accurate, or a couple of days beforehand is probably fine. One of the challenges with satellite imagery is, cloudy days in the spring, and are you getting a measurement from the day you killed, or is your measurement from 10 days before you killed? And what happened during that 10 days of cloudy weather in terms of the cover crop nitrogen potentially increasing? That is one challenge I foresee with using satellite imagery versus ground-based NDVI measurements.
Any final questions for Charlie? Here we go. Nicole asked if you have any suggestions on how to best help farmers get correct sand and clay percentages, and how much will guessing those percentages cause error in the calculation? Yeah,
so the sand and clay is a relatively sensitive part of the model. And what I recommend is for best performance to get a soil sample measured, from the lab. So like the Penn State Ag Analytical Services Lab, one of the custom services you can get when you submit your soil sample is texture analysis. So if you're sending in a routine soil sample, the next time you do that, check the box for texture. And I think it costs $20. The thing about texture is it's never going to change. So that's a one-time investment that you can make. And maybe if you've got like a contract with NRCS for EQIP funding or CSP funding or something like that, I'm sure you could get that additional cost of soil sampling covered in a contract like that from NRCS. I have seen relatively good agreement between the NRCS soil survey maps and our on-farm sites. When I've compared them side by side, they're usually within a few percentage points of each other. So I think that would also be an alternative way to get started in the ballpark, is to look up your soil survey map. And that's going to get you in the ballpark. And if you want to get closer, then, submit a soil sample to the lab.
Great, thank you. And then finally, Kristen asked, how expensive are those handheld NDVI units?
Yeah, so when I bought my Greenseeker sensor, probably 6 or 7 years ago, it cost $500. Over time, the cost has gone up. And then one thing that actually happened is the company that makes them, Trimble, they decided to create a new version of their sensor, and they stopped making the old version. And so the old version is completely sold out, and the new version isn't on the marketplace yet. And so there's sort of a gap in that. And I'm sort of waiting to see what their new sensor looks like, how expensive it's going to be, use it side by side with the old sensor to make sure they're giving the same numbers. So we'll have to see on that.
It's super helpful. Thank you for answering these great questions. Thank you for presenting this great work you and your colleagues have done.
Yeah, it's my pleasure. I'm glad to have the opportunity to share, and look forward to working with folks in the future on this. So thank you.